Once you have your program of choice open, in this case Genome Compiler, we then need to import the sequences of interest to be compared to one another. Since both human and Neanderthal data can be found in a public gene repository called NCBI, we can directly upload it from there. Alternatively, the genomic data can be directly imported to the program if it cannot be found in such repositories. At this moment, I would like to point out that we are comparing the mitochondrial DNA of modern human to the mitochondrial DNA extracted from the Neanderthal specimens in a similar manner to how Pablo would have. Once the data is imported, it then needs to be put into an environment where it can be compared. A new project file is opened and the reference sequence is then added. For this comparison, we are using the human genome as the reference sequence. Once the reference sequence data is loaded, a sequence alignment can be performed and the comparison sequence is then input. Here we are using Neanderthal data as the comparison sequence. Once the alignment is started, a program called Clustal is then run. It aligns multiple sequences and generates comparison data. This process may take some time. The finished result is a quality control alignment which details mismatch content and at what location the mismatches occur. To better illustrate this, a side-by-side view is created which allows us to select a mismatch and go directly to it in the sequence data. The letters highlighted in red are base pairs that differ from the reference sequence at that location. Due to the age of the sample, the sequencing is imperfect. There are gaps in the genome that can be seen in visualizing the sequence data. This data allows scientists to infer phylogenetic relationships and help to answer questions in evolutionary biology.